Good afternoon, everyone. I'm Dr. Wheat, Associate Professor of Biology at Jefferson Community and Technical College. I'm glad to be here to speak to your research program this afternoon, and I'll be talking about learning and literacy. So, to tell you a little bit about me, I have a bachelor's degree in biology from Indiana University, and from my bachelor's degree, I went straight into a PhD program at the University of Cincinnati College of Medicine. My dissertation research was on cystic fibrosis in tracheal epithelial cells. My first full-time college teaching position was at Gateway Community and Technical College, which is part of the same KCTCS system as Jefferson. And so I moved from Gateway to Jefferson in 2009, and in 2013 I was promoted to associate professor with tenure. So here's some topics that we'll cover today. We're going to talk about learning. We're going to talk about some metacognition and reading processes, and then we'll talk about visual literacy. So let's get started. So we know from human anatomy and physiology that there are cells in the nervous system called neurons. We know these cells take in information at the dendrites, and if there's enough stimulation, an action potential will happen, and they can transfer that information to what's called a postsynaptic cell at this synaptic terminal at the end. Okay? So we know that these neurons can take in information, respond to a stimulus, and they can pass on that information. The arrows show this one-way movement of information from the dendrites to the axon. And in biological systems, this information is really the movement of ions across the cell membrane. Ion channels open because of a chemical or a change in voltage. So we learn the mechanics of this in anatomy and physiology in Bio 137, A&P1, when we talk about the nervous system. Okay? And if we blow up that synaptic end bulb, we can see the details of how this neuron communicates with whatever this postsynaptic cell is. And in Bio 137, you'll get the details of that. The details of that are not terribly important right now. But what I do want to talk to you about is how learning how these neurons work, learning how these chemical synapses work, can help you to understand how the brain works as far as taking in information, remembering it, retrieving it so that you can use it later. That whole process of learning with all those steps that are involved. So think of something that you've learned to do. Think of something in your lifetime that you've learned how to do. Have you learned how to drive a car? Have you learned how to play an instrument? Have you learned how to perform a sport, cook, train your dog, walk a certain pathway to go to the grocery store, right? What is one thing that you've learned to do in your life? Okay. If we were doing this as kind of a live interactive session, I would recommend that students kind of give me some feedback on that, right? Tell me some of the things that you're thinking about that you've learned to do. Then we can talk a little bit more about how did you learn to do it? What are the steps that you went through? And then the other piece that I want to talk to you about is why did you learn it? What was your motivation? Did someone make you? Did you want to? Was it to avoid negative consequences, or was it to pursue positive consequences? And then as you begin to get into your courses that you're taking, again, think about what your motivation is. Sometimes it's a required class that you have to take, and so your motivation is doing as well as you can to fulfill that requirement. But sometimes it's a topic you're really interested in, and so your motivation may be your curiosity and wanting to know more about that topic. And sometimes it's both of those things at the same time, right? We can have multiple motivations for why we want to learn things. So, Dr. Rita Smilkstein is an education and a researcher, and she's published a book called We're Born to Learn Using the Brain's Natural Learning Process. Okay, and here's a link to a PDF if you'd like to read more about her handouts. She does a lot of training for teachers, for how teachers can understand how learners learn in their brain, and then how teachers can design curriculum for that process. And so this is from that handout that she gives to teachers. And she talks about these stages of this natural human learning process. And the very first stage, what we were just talking about, is motivation. So unless you're motivated, you're really not going to learn something very well. And the motivation may just be avoid something painful, negative consequences, right? But the more you can move toward positive motivation, interest, desire, curiosity, the stronger the neural connections and the better you're going to be able to remember the information that you're learning. Then we go through some stages of practice. Okay? Beginning practice, more advanced practice, and eventually we get to a stage where we feel pretty skillful. We refine those skills, and then we can sometimes even get to a mastery level where we can teach things to other people. It feeds into other interests, and sometimes this kind of becomes pretty consuming. Once you reach the mastery level of, for example, piano playing, you may be a concert pianist by then, and then it's a significant part of your life. If it's playing basketball, once you get to the mastery level, even if you're not a professional NBA player, you may find that you're still playing on a league or doing a lot of pickup games. It takes a significant portion of your life that you want to spend toward that thing because you have that mastery, you have that sense of enjoyment, that it's easy to do this thing that you've learned and it's enjoyable to do this thing that you've learned. Right? So that's eventually where we want to get to, is that stage of mastery. This is another figure from Dr. Smilkstein's handout that she gives to teachers. And look, she talks about growing dendrites is what learning is. And we were just talking about those dendrites are part of that neural connection and so important when you're trying to learn and then retrieve what you've learned from your brain. Okay, so she draws here a cell body kind of in a triangular format, and then she shows some of these extensions. She shows how they can branch and then how they work together for one cell to communicate to the other cell. So where we were just talking about the axon terminal communicates with a postsynaptic cell, that's exactly what she has drawn here. And learning over time, the actual mechanism of learning is growing these branching structures in the brain. So if these are the kinds of dendrites that we have at birth, then they get significantly more as the brain learns and grows. And then we can even do some remodeling, right? You can imagine that it can't just happen exponentially forever because the brain eventually would be so taken up with dendrites there'd be no room for anything else in the brain, no blood vessels or anything like that. So at some point we start to lose some of these neural connections in favor of making others stronger, and that's the use it or lose it phenomenon, right? If you've learned something and you use that information over and over, then it's easier to recall. The information that you don't use over and over, you forget because your brain is remodeling to really preference the neurons that contain the information that you're using for recall over and over. Okay? And so where that really starts to come into play is in studying, we really recommend that people do what we call retrieval practice, which is remembering what you've learned in various and different ways. So when we get to what do the most effective students do a little bit later in this presentation, I'm going to talk about something called retrieval practice and the fact that what the most successful students do is answer questions. Seek out and find sample questions, answer those questions, because the process of answering questions does all of this use it or lose it, helps with retrieval, helps with remodeling in the brain. Okay, so Dr. Smilkstein has come up with the 5 rules of the brain. Dendrites grow only from what's already there. Dendrites grow for what is practiced. Dendrites grow from stimulating experiences, use it or lose it, and emotions affect learning. Okay, we can really delve really deeply into these 5 rules, but they kind of make sense based on this anatomy that we've been talking about and the physiology of how the brain works, right? So dendrites grow only from what's already there. So one of the most effective things you can do is start to activate the neurons that have the information that we need to grow from. Instructors call this activating prior learning. And there's a lot of different ways that they'll do it. At the beginning of a new unit of information, like a new chapter, they may give you a summary. In a face-to-face class, they may say, get out a sheet of paper and write down everything you remember about enzymes, because we're talking about the digestive system and there's a lot of digestive enzymes. We need to activate your prior learning so that you remember everything that you know about enzymes, and make sure that you have a complete story about enzymes, because that's what the digestive system does, right? And so there's a lot of ways that you can activate prior learning, but that is to help activate the neurons that are going to grow new dendrites and grow new neural connections. Dendrites grow for what is practiced. So practice over and over again. You want spaced practice. You don't want to cram everything in the night before. You're really going to want to space it out because these dendrites take some time to grow. You especially want to make sure that you incorporate some sleep in between when you learn something and when you need to remember it, because these dendrites tend to grow while you're sleeping. Dendrites grow from stimulating experiences. So you may notice instructors starting to really incorporate active learning into their pedagogy or their teaching techniques, right? Because just you're sitting here watching this video right now. This is a very passive process. It's very difficult in that asynchronous way for me to really stimulate. But if you're taking some notes, if you're writing some things down, if you're talking to other people after you watch this video about what you learn and remember, those are all more stimulating experiences than just passively viewing a video, and they will help you remember the things that you're hearing and learning by stimulating these neurons and these dendrites to grow and make connections. Okay, that's often why in a lab we recommend to students that you kind of get up and move around the room to interact with the different models that are there. That's often why we'll put you in conversation with other students so that you can talk about the different things that you've learned, because those are all more stimulating to your brain than just passively taking in information. Just a passive input may not help you grow any new neural connections at all. It's the interactions and the stimulation that will help you to grow those new connections. We already talked about use it or lose it, right? And emotions affect learning, right? So you want to try to maintain your brain as a happy, anxiety-free space. That's often easier said than done, right? And so just to be aware of the impact that anxiety has on learning is a thing to be conscious of. That's an entirely separate presentation that we could do that talks a lot about the specific neurotransmitters that get released, specific other chemicals and molecules that can modulate that process. But ideally, you will try to learn in as calm a way as possible, because emotions do affect your ability to learn. So here's some major points about learning for students from Dr. Smilkstein. And these are things that we've already kind of talked through. Learning takes time because you need to grow and connect dendrites. Self-doubt and fear prevent you from learning. So confidence, interest, curiosity will help your brain to learn and think and remember. So there's another author that we can talk about, John Medina, who wrote a book called The Brain Rules. And if you look at these are exactly what Dr. Smilkstein talks about as well. She talks about repeat to remember and remember to repeat. So he talks about that use it or lose it kind of thing. We are powerful and natural explorers is a rule that he added. He originally started out with 10 rules, and then he's up to 12 brain rules now. But this is the curiosity piece, right? That the fact that emotions can really impact learning, and curiosity is a really powerful way to bring in motivation and to help. He emphasizes sleep. He emphasizes low stress environments and the sensory integration. So these brain rules fit very closely with what Dr. Smilkstein also teaches in her handout. So what do we do in the classroom? Activate your prior learning, I just talked about. Activate your emotions. Really go deep into why are you trying to learn this and how will it help you, and hang on to that motivation that you have. Repetition is key. How can you repeatedly practice what you're learning? How can you apply it in different contexts? Each new context that you apply information in can grow some new branches of dendrites and neural connections. And then what does your brain need in order to grow? The same stuff that we tell small children, that we tell elementary children, that we tell you in middle school and high school. You need to get a good night's sleep. You need to eat good food. You need to exercise, and you need to have some time off, right? So you really need to try to space your practice. And I don't mean space like cram once a week for your online class. Do it all in the few hours before the deadlines are, Sunday at midnight or whenever your deadlines are. By spaced, we really mean to space it out throughout the week and throughout the semester, and really make yourself a plan. And when we get to what do the good students do, planning is one of the things that we'll talk about. Okay, so that's a little bit of a tour through the brain and some researchers of the brain and what they say that you need to do to take care of your brain, because your brain is where all this learning is going to take place. Now, your brain does thinking. And so the next thing we're going to talk about is we're going to talk about some things related to thinking. And specifically, metacognition is thinking about your thinking, monitoring your thoughts to see if you're confused, if you're in a place of understanding, and what you can do to help yourself to better understand, especially when you're reading. So when I do this seminar with live students, I often ask, do you like to read? Drop in the chat a yes or no. So just think to yourself kind of how you would answer that question. Do you like to read? And then if people say no, then the next question is usually, why not? And that's a thing to think about. You can think a lot about your history as a reader, and you may come up with specific experiences you've had that influence whether or not you like to read. Sometimes when I do this in a class, I have students kind of work together in groups to talk to each other about some formative experiences around reading that they've had. But then I also talk to them about the fact that you can have a growth mindset around this. You can employ your growth mindset to say, okay, these are the experiences that I've had, and I want to grow even more as a reader. I either want to overcome some of these negative experiences that I've had, or I want to employ even more positive experiences to learn to be a better reader, especially in the context of college level courses. One thing to think about is, are there certain things that you like to read more than others? and think about why those things are more comfortable for you. And think about ways that you can use that comfort zone to learn to be more comfortable with reading, especially with reading at the college level. So when you read, do you ever get confused? Is there ever something that you've read before that you're like, okay, I read that whole paragraph and I don't even know what that said? So that's what metacognition is. Metacognition is realizing when you're confused and then knowing what to do to help clear up that confusion. There are strategies that you can use to problem solve. You can become a problem solver as a reader, and so you can realize that everyone has points when they get confused or they realize they don't understand what they're reading. And they use different strategies to get back to a place of understanding and comprehension. And we can learn those strategies and use those strategies to become a better reader or to become more comfortable with reading. So we can talk through some of those strategies. What I want to demonstrate for you is something called a think aloud. And I'm going to demonstrate for you some of the metacognitive strategies that happen when you're reading a text that's unfamiliar. Okay? So when I usually employ this, it's in either a general biology class or human anatomy and physiology class. And it's usually a reading that I've had students do to prepare for the lecture or a live online class session in an online class. And so here is a paragraph that is towards the end, and I can use it just as an example of how I think about reading while I'm reading, those metacognitive actions that we can take. Okay, so it says growth and development. Every different species of living things has a particular pattern of growth and development. Okay, so it says growth and development here, and it says growth and development here. So growth and development seem like they're two separate things. Okay, growth and development may seem like very similar terms, but they're actually quite different. Okay, so I did some predicting there that panned out. All right, so I was right. They're quite different, so I guess they're going to tell me how they're different. Growth simply refers to the increase of size and mass over a period of time, and growth occurs... Okay, hang on. Growth simply refers to the increase of size and mass over a period of time. Okay, so again, size and mass are going to be different. It increases in both size and mass over a period of time, and growth occurs through a cell division process known as mitosis. Okay, during mitosis, we're still talking about mitosis here, one cell splits into two duplicate cells, adding mass to the body. Adding mass to the body. One cell splits into two duplicate cells, so that's probably increase in size. And then since two cells are heavier than one, adding mass to the body? I guess that's right. This process allows all body parts to grow the size appropriate for the individual. Okay. Development. So we're switching from growth to development. Okay. Development is a broader term for the transformations or changes in an organism throughout its entire lifespan. Broader term for the transformations or changes in an organism throughout its entire lifespan. Okay, so I think they're talking about like humans, like first you're an egg and then you're a fetus and then you're a baby and then you change over the span of the lifetime. Okay. For example, a moth has several developmental steps during its life cycle, such as egg, larvae, cocoon, and adult moth. A human's development includes fetus, baby, child, and adult. Okay, once again, I did some predicting and it panned out for me. Viruses. So this whole reading was about viruses to begin with. And here's where they're bringing viruses in. Okay, viruses use host cells by manipulating them into building new viruses. Each individual virus is created in its fully formed state and will neither grow in size or develop within its existence. So this original reading I was using to teach a little bit about viruses. We're all interested in learning about viruses because of novel coronavirus SARS-CoV-2. And this particular reading gives all the characteristics of living organisms and asks, are viruses alive? Right? And so one of the characteristics of living organisms is growth and development. But then based on these last few sentences, viruses don't grow in size and they have no development. They're just formed and they're the same size, they're the same mass. They aren't built of cells that divide, right? They're just formed as like a protein and a DNA or protein and an RNA, and then they can go and be formed again by cells. Okay? So that's a think aloud. And when I do that in front of students, I always ask, what did you notice? What did you see? When I do this in a live online class session, I will always tell students, what did you observe? And we will write down types of behaviors that I exhibited that people observed. If you want to pause the video, you're welcome to go ahead and write down some things that you noticed that I did in that think aloud when I was trying to say out loud all the things I was thinking while I was reading that paragraph. So in reading, you can monitor your understanding. You can monitor your comprehension, and then when you deviate from that place of understanding and comprehension, there are strategies that you can use to bring yourself back. So here are some fix-up steps. If you notice that you're confused, you can ignore the unclear part and read on to see whether it gets clearer, or maybe the unclear part turns out to be unimportant. So when I first read that title, Growth and Development, I was kind of like, I don't even know. Do I understand that? Do I not? I don't even know. Let me just keep going and I'll see if I get confused. You can reread the unclear part more carefully. You can reread the sentences before the unclear part. So you know that you're going to hit this point of confusion. Can you go back and read a little more carefully and see if you missed something that will help you continue to be understanding in the understanding mode of what you're seeing? You can try to connect the unclear part to something you already know. And then you can always get outside help from peers, from the teacher, and from resource materials. So in one of my live online class sessions, as we were working on talking about reading skills, I had students read a really kind of difficult part of a textbook, very short, but a little bit complicated reading, so that then we could try to do some of these fix-ups and go back and try to understand it, right? And I had told students, this is a pretty difficult reading. It's possible that you will have lots of areas of misunderstanding and confusion, but then we can work on it to clear it up. And one student recommended, if you go back to the lab book and read a similar section in the lab book, that might help us with reading this reading, which I thought was a really great suggestion. And another student said that they hesitated to do that because what if they were confused on that reading too? They weren't seeing how possibly using resource materials can clear up the confusion on this more difficult reading. They thought that just adding more reading would just add more confusion. Okay, so there are times when some resource materials may be helpful to you in clearing up your confusion. Some other steps that you can do. This is a bookmark that you can use. This has some of these metacognitive processes that you can use while reading. You can do some predicting. You can do some visualizing. What do you picture? What do you see? You can ask questions. You can try to make connections. When you realize that you're confused, you can identify the problem. You can talk about, I got confused when I hit this part. I'm not sure about this area where these numbers are. I would actually have to look at that in a little more detail. I didn't expect to have this really complicated chunk of text, and I might need to spend some time going through it to figure out what it means. Using the fix-ups that we just talked about, either rereading or continue to read and then check back to where you need to. Summarizing is sometimes helpful. And so when we talk about successful students, there are some specific strategies that students employ, both while they read and also just while they go through a semester and do any kind of the work related to a course. And so one of the first things that students do is they try to plan and organize. That involves setting goals, previewing or skimming assignments to decide how to approach them. So when you know you have an assignment coming up, you should go ahead and take a look at it. It might not be due for another week. You might think, oh, I'm going to work on this next Thursday, but you should go ahead and take a look at it. Because if you wait till Thursday and you open it up, and something that you thought you could get knocked out in an hour looks like suddenly it's going to take about 6, that's going to mess up your plan. Especially in like Bio in 37, when we get to muscles and bones, there's quite a bit of information to take in. And so up until then, you've been like, oh, we talked about tissues, and that wasn't too bad. Oh, we talked about skin, that wasn't too bad. And then we hit skeletal system, and you're like, this has more information in it than anything else I've studied so far. Yes, it does. There's hundreds of bones in the body, and we're going to learn a select few of those, but we still have to learn them and learn them in some detail, right? So making sure that you open assignments, that you know what's coming up so that you can plan, is going to really help you with budgeting your time and doing your time management. Oops. Successful students create a timeline to divide big tasks into manageable chunks. And that chunking is one of these metacognitive processes and is one of these success strategies that fit with that spaced practice, right? Successful students monitor their own work. They can check their progress against their timeline. They can troubleshoot if there's any issues. And they can ask themselves if they're doing their best work. Okay. There are times where you can kind of do the minimum for the expectations that are required of you, right? On a reading assignment, if you just have to make 3 comments, then you can really just make 3 comments, right? But at some point, you are going to want to try to get as much out of your coursework as possible, especially if it's a course that's a prerequisite to another course, right? So a lot of students in the summer research program are taking like a chemistry to build to another chemistry, or physics to build to engineering classes, or biology to build on another biology class. If you're going into nursing and you're taking Bio in 37, you then have to take Bio in 39, and then Bio 225. All those build on each other. And then when you get to nursing, they do actually expect you to remember what you've learned, right? So there are times that you can do kind of the minimum to meet the requirements of an assignment. And then there are times that you really are wanting to try to build as many neural connections in your brain as you can, so that you'll remember this for 4 or 5 semesters from now when you're in the nursing program and you need to retrieve that information. Successful students direct their own learning. They can pay attention to when they've lost focus, and they can use some of these metacognitive strategies to kind of get away from confusion, get back into a place of understanding. They try to create an optimal studying or learning environment, and they can advocate for their own needs, right? So they know what it is that they need to learn, and they can direct themselves in that learning. They can try to create a schedule or a routine that's helpful, and they can use an environment, right? If you really need it to be quiet and it's not quiet at your home, then reserving a room at the library and going and sitting in a little room in the library. Have your standing appointment with the library for all 16 weeks of the semester if you know that that's something that you need. And then successful students do a lot of self-reflection. This is part of this metacognition where you're reflecting on your own thinking and your own learning. Okay? Successful students can assess about how a task went, consider how effective their strategies were, and figure out the changes to be made the next time around. So in some of my classes, I offer students a practice midterm before the midterm that counts for points. And I've had students just blow off the practice midterm before because it doesn't count for anything. But it does count for things. It might not have points assigned to it, but it does help you figure out if your computer can run the midterm exam. It helps you figure out where the controls are for the midterm exam. It helps you figure out where the submit button is and where the timer is and what it's going to be like to flip through the questions and answer the questions in the actual midterm exam environment. The students who do the practice exam do better on the midterm. It might be that those students were going to do better on the midterm anyway, and those are conscientious students who do all of everything that's on their plan, and on their plan was the practice midterm. It may be that if you're so out of what's going on in the class that you missed the practice midterm, that you're not even ready for the regular midterm, right? But also there's probably a little bit of students for whom the practice midterm really set them up for success in the actual midterm by alleviating some of that anxiety because they've already been through the process. They took this entire test that had 0 points attached to it. So there's no anxiety, there's no performance anxiety. You can click around, you can move forward and backward, you can test out the buttons and see what things do, right? You can really play around with it with 0 consequences and get really comfortable with it. So then your anxiety level is much lower when you actually go in to take the actual midterm, right? And isn't that great to be able to feel that much better and regulate your emotions around learning? So why do I teach this? Why did I do this think aloud? Why do I present this to research students? Metacognitive strategies can be taught, and they are associated with successful learning. Successful learners have a repertoire of strategies to select from and can transfer them to new settings. So a lot of what I've said today probably isn't new to you. You've probably heard these things before and maybe even heard them in high school. But it may not have occurred to you that these are tools in your toolkit that you can apply to the classes that you're taking at the college level. They'll apply to classes that you take in nursing school, at the higher undergrad level, or even in graduate school. Okay? This researcher found that few college instructors explicitly teach strategies for monitoring learning. They assume that students have already learned these strategies in high school. I taught this way for a very long time. I just assumed that you'd already heard this, that you already know this, and that you're applying this on the regular basis. But sometimes it does help to have a little bit of a refresher or to put things in a different context. Many students have not learned these strategies in high school. They're unaware of the metacognitive process and its importance to learning. Rote memorization is the usual and often the only learning strategy employed by high school students when they enter college. Rote memorization is not going to get you far. In science classes, there are facts to memorize. There are different factual things that you will need to know, names of structures and where they're located, right? But you're very quickly going to need to move up Bloom's taxonomy or the levels of critical thinking to where you can analyze new information with what you've been told or apply the information that you've learned to a new situation. Okay? And so that's one of the reasons why I take the time to talk about metacognition, to do the think aloud, to really talk ourselves through the steps of planning your learning and monitoring your learning. Things that you've probably heard before, but it always helps to hear again and to think very critically about how am I going to apply this to my classes this semester. So we've talked about the actual mechanism of learning, growing new dendrites and the conditions that help with that. We've talked about thinking about your learning, both when you get confused and how to clear up confusion, as well as overall metacognition over the course of a semester. How do you plan and monitor your learning and assess what you need to do differently to continue to learn and grow over the course of the semester? So the last thing I want to talk about is one specific skill called visual literacy. And this is basically how to read figures and artwork in your textbook and your other readings. So many students tend to skip over the artwork in their textbook or their reading materials. But this artwork is specifically designed to help you understand the concepts that it takes paragraphs to describe in the text. And so part of your metacognitive strategy may be, okay, I read it and I reread it. Maybe I need to look at the figure to see if that helps, right? So you're going to want to try to get comfortable figuring out what the artwork in your textbook is trying to explain. And my biggest piece of advice to you is figure out what the arrows mean. So information can be presented on a coordinate plane. So that means sometimes information is presented in a graph with an XY axis. So here the x-axis is time or the progress of the reaction, and the y-axis is increasing potential energy. So the higher something is along this direction, the more energy it has. This figure also uses color coding and it gives you a legend. The red line is activation energy needed without a catalyst, and the blue line is activation energy needed with a catalyst. And so the red line is here and the blue line is here. And so if the red line has this much energy, this blue line has this much energy, so it looks like the catalyst lowers the amount of activation energy needed. And in biological systems, those catalysts are enzymes, and that's exactly what happens. Enzymes lower the activation energy of the reaction. Information can be presented cyclically. The arrows here show the sequence of events, and they occur in a cycle. One of the things that I always recommend to students is you want to read the captions, especially if they have numbers. They're showing you a sequence of events. So here's the enzyme. Here are the substrates, and the substrates bind, get converted to products, and the enzyme itself is unchanged, right? So we bind more substrates, convert them to products, and the enzyme goes on to bind more substrates, convert them to products. The enzyme's unchanged, substrates, products, enzyme. Okay? And this sequence will continue until we run out of substrates. Okay? If there's nothing else to bind to the enzyme to get converted, then the cyclical reaction can come to an end. And that enzyme just hangs out until we have more substrate. Here there are several different arrows. Okay? So the first thing to notice is all these ribosomes aren't there all at the same time. This is a single ribosome, and it's showing a sequence of events, and it's showing the passage of time. So this happens, and then this happens, and then this happens, okay? And then eventually that ribosome moves off because the completed polypeptide makes it into the lumen of the rough ER. Okay, so the arrows that are on the top of this pink mRNA, these arrows are showing that this is a sequence of events. This leads to this, leads to this. Okay. The set of arrows below that pink mRNA are showing things either binding or unbinding. Okay, so what happens is this polypeptide and this signal recognition particle come together, and the SRP is bound to the signal peptide. That binds to a receptor, which will put this ribosome in place to begin feeding that protein into the lumen of the ER, where it will finish folding. Okay. If the figure really is trying to represent a bunch of ribosomes all attached at once, it would omit these arrows, and it would demonstrate more clearly that everything was happening at the same time. Now, information in a figure can also be color-coded. So the blue and purple is the sugar phosphate backbone of the DNA double helix. And there are 4 bases, and so they give each of the bases a different color, and they give you a key right here so that you can understand, even though they're all up here and also labeled underneath. Okay, so that we could edit this and just take this part of the figure if we wanted to, and we would know that yellow is thymine and green is adenine. But then the key also tells us when we widen our view a little bit, when we step back and look at the double helix as a whole, A still pairs with T and C still pairs with G. Okay, and so they're representing a lot of key information in this one figure. They're showing you the sugar phosphate backbone. They're showing you that the A and T bases make 2 hydrogen bonds, where the C and G bases make 3 hydrogen bonds. And that convention is even carried down here. A and T have 2 hydrogen bonds. C and G have 3 hydrogen bonds. They point them out and label them, that those dotted lines are hydrogen bonds. They point out here that we have the phosphate group and the deoxyribose sugar, so this is a sugar phosphate backbone. So as we've seen, a figure that uses symbols will usually define those symbols in some way, right? Our original graph that had on the coordinate plane had red and blue arrows. It defined the red and blue arrows. Just now, when we were looking at that figure with the bases, it defined the color of the bases. If you see a figure that seems complicated and you see symbols that aren't defined, you may have to look at some prior figures. So in the next example, the symbols shown in the center of each structure in the nucleus of the atom aren't defined in this figure because they were defined in the previous figure. So you may have to go back to that previous figure to understand the symbols, because this textbook carries those same symbols through the entire chapter and even into later chapters. Okay, so in our anatomy text, nerves are always yellow. And so anytime that you see yellow, you can pretty much guarantee that it's a nerve, and look for the label to tell you if it tells you which nerve, because the book establishes early on when talking about nerves that nerves are going to be yellow. Lymphatic vessels are green. Arteries and veins are red and blue. So there's a lot happening in this figure. We use this figure kind of to demonstrate, but also as reference. So later when we're like, okay, what was nitrogen's atomic number? It's right here. So this is both for demonstration and reference purposes. Okay. In each of these nuclei, there's P plus with a number in front of it. There's N with a kind of a 0 next to it with a number in front of it. Those numbers are going up. But what if you don't know what P and N are? P and N are defined in the previous figure, where it shows a couple of ways that we can denote atoms. Here's the electron cloud model. So all the electrons just kind of circulate in a cloud. But for the most basic atoms, the electron shell model works. So for the atoms that are involved in human anatomy and physiology and human biology, we can use this electron shell model of the atom. Now, the chemists kind of laugh at the biologists because this kind of model only holds true for just the first couple of rows on the periodic table. After that, you have to have a whole different way of visualizing the structure of the atom. But we don't have to get that far in biology because we're just using pretty much the first 2 rows of the periodic table, right? So chemists are like, oh, you need to teach this more sophisticated model of the atom. And we're like, this is called the Bohr model of the atom. And I'm like, no, I can stick with the Bohr model of the atom. It works for my classes. And in this model of the atom, the protons and neutrons are in the center of the atom in the nucleus, and the electrons circulate around the nucleus in these areas called electron shells, right? It's kind of like the shell on the outside of an M&M candy, right? The colorful shell. In the legend to this figure, they point out protons, and they say we're going to abbreviate protons P+. Neutrons, we're going to abbreviate N with a 0. Protons have a positive charge. Neutrons don't really have a charge. We say they're neutral because they don't really have a charge, and that's why the 0 is there. And then electrons will be abbreviated E with a little minus sign because electrons have a negative charge. So by looking at this figure and understanding this previous figure, you can then start to understand some of the abbreviations that are used in this, which is just the very next figure in the textbook. Don't just skip over figures in the text, especially if they seem hard to understand at first. The struggle that your brain experiences as you work to understand the figure is the process that you need to go through in order to grow new neural connections. Okay? Some people even say you have to forget in order to remember. You have to forget in order to learn, right? Confusion and forgetting are key steps in the learning pathway because that's where you're starting to activate the neurons that need to grow those new connections, those dendrites, and those new connections between neurons. Right? So struggling to understand something is actually a good thing because you're likely, once you get to that point of understanding and remembering, then you're going to be more likely to be able to access that information and use it to application or analyzation kinds of questions. Other ways to work with figures other than just trying to read and understand them, maybe try to redraw the figure from memory and then look back to see what you missed. This will tell you what you retained and what you didn't, and will help increase your learning. You often can't really get everything out of a figure just by staring at it. You usually have to interact with it in some way, and trying to redraw from memory is a really good way to be very interactive with that figure as you're trying to understand what it means. See if you can explain the figure to other people. You can up the challenge by covering words with sticky notes, so you have to remember the labels. You have to remember what things are called. Practicing explaining the figure helps to put the information in your own words. If you can explain the figure to someone else, then you really have an understanding of what that figure is illustrating. Pets are really good for this. My dog loves it when I talk to her, no matter what I'm saying. So if I'm practicing trying to smoothly describe something, She just wags her tail and listens and loves the attention. Kids are good for this. You can be like, hey, let me practice this on you. Let me try to explain this to you and tell me where it does and doesn't make sense. Tell me what I need to work on. Okay. But you can do it, just put yourself in a quiet room with nobody else around. Can I put this in my own words? Can I start to explain this? Where do I get stuck? Because that's the metacognition piece. And what can I do to clear up my confusion? Those are the fix-up steps. So in summary, we've talked about some things your brain needs to grow and learn and what that mechanism is. And that mechanism is the growing of dendrites and the increased neural connections. Then we talked about literacy, and we talked about it in two ways. First, we talked about reading and metacognitive strategies that you can use while you read. And then we talked about visual literacy, or reading the figures, right? But those same metacognitive strategies will work whether you're trying to read text or read a figure. And those metacognitive strategies are designed to help you grow those neural connections. So I'm hopeful that this was helpful for you in some way to think about how learning happens and how we can best have our environment be conducive to those dendrites growing, how we can tackle reading, how we can tackle visual reading, and how we can use metacognitive strategies to help us plan, to help us monitor our understanding, to help us reflect, and then reiterate that cycle again with more planning, more monitoring our learning, and more reflection as you go through all of your courses in your program of study, and as you become more and more successful, as you pile up those wins, right? If you have any questions, please feel free to contact me by email. I appreciate the chance to come and talk to you today, and I'll take any questions that you have.